Who are the hosts?
Very good morning.
Good morning.
Dr. Parvati Madam Garu, Head Department of Botany, Government Pingili Government Women's College Autonomous, Varangal. And Sarita Madam Garu, good morning.
Good morning, sir. Good morning, sir.
Morning. Madam Bhavunara.
Good morning, sir. Good morning.
Madam Bhavunara. Sarita Madam ji.
Morning, sir. We're Bhavunara.
Bye Madam, bye.
Sir, I want to introduce you, sir.
Madam.
I want to introduce, sir. Very
Okay,
good
madam.
afternoon to all.
Okay, madam. Please you proceed.
Okay, sir. Very good afternoon to all.
Good morning, sir.
Padala Thirupathi,
Madam.
sir.
Madam, good morning.
Padala Thirupathi, sir. Sir, academic qualifications are MSc BEd, set in life sciences, MEd UG set, then MA psychology, also having a set, MA English, PG diploma in graduation, PhD in Kakadi University. Sir, teaching experience is 17 years and the research experience is 12 years. Sir, additional qualifications additional responsibilities in the college is NSS program officer, head department of the botany, coordinator Haritaharam, convener Hippo Club, convener of DOST. The training program attended human value professional ethics basic in IIIT Hyderabad, the human values and professional ethics advanced in IIIT Hyderabad, then answer in Usmania University, then was in Manu Hyderabad, then Mahatma Gandhi Rural Development Program, Energy Varangal. The invited talk as keynote speaker through online mode. Sir, given 2 lectures is the keynote speaker. One is the evolution of sporophyte in Bryophyta and stellar evolution Pteridophyta. Giriraj Government Degree College, PG and UG, Nizamabad. Then another speaker, biofertilizers and organic farming for sustainable agriculture, GDC, Gambiraupet. Sir, he is also conducted national level certificate course, conducted 1 month duration, that is. Another one is 1 national level online coaching in plant sciences. Sir, conferences attended, international conferences 37, national conferences 107, then workshops is 8 and FDPs are 20. Sir was presented papers, 2 international conferences, 2 papers are presented, then national conferences, 2 papers are presented. Then sir, paper publications are, international paper publication is 7, the national publications are 4. This is a brief introduction of the Padala Thirupathi, sir. Very good CV, sir. Viewers, very nice. Congratulations, sir. I request you to kindly give a lecture, tissue culture and biotechnology application, sir. Please give, sir.
Tirupati sir,
Thank you very much, madam, for your nice introduction about me. Now, I would like to, before going to start the session, I would like to share a few words regarding of today's seminar. First of all, I would like to congratulate the Department of Botany, Government, Pingili Government College for Women, Autonomous, Varangal, and the Department of Botany, Head Department of Botany, Dr. D. Madam Garu, and Sarita Madam Garu, and other faculty members of the Department of Botany. And today's today's seminar chairperson, Dr. Rajesh sir, and other faculty members of the Government College Autonomous, Women's College Autonomous. So wholeheartedly thankful to you for giving me this wonderful opportunity as a resource person of this today's seminar. So I know about you, and because of your elderly, the Varangal, Madam Garu, and Parvati Madam Garu. Both of they are very well known to as a department of botany lab mates. At the same time, my senior faculty members and today's resource person, Dr. Savita Madam Garu, and my colleague earlier, Dr. B. Sandhya Rani Madam Garu, GDC Lakshadipet, and also thankful to each and every faculty of today's seminar. So, my dear, sharing of today's my views on lecture on today's seminar, that is plant tissue culture biotechnology. I hope the slide is visible.
Yes, sir. It is visible, sir.
Okay, thank you. So, by today, great day because of English College, Government College is giving me full opportunity. That's why I am very happy to share my knowledge in today's seminar on plant tissue culture and biotechnology applications. Myself, Dr. Padala Thirupathi, Asst.Professor, Department of Botany, Government Degree College for Women, Jagitial. Okay. So, I would like to share my knowledge regarding of today's seminar. So, my area, today's, I'm going to deal today's seminar on applications of biotechnology. So, before going to that applications, what is biotechnology and how many types of the biotechnology which we are observed in the present scenario. So, biotechnology is the applications of science in which we study the use of organisms, biological process, or systems to manufacture products, aims to improve the quality of human life. So that is the biotechnology. So biotechnology, the applications of science in biological process, the systems to manufacture products, aims to improve the quality of human life. So that is biotechnology. And how many types of the biotechnology which we are observed? So first one, we will going to discuss about the green biotechnology. So green biotechnology, it is the applied to the agricultural process. In the field of agriculture, so we are called green biotechnology. The three main areas of applications are plant tissue culture, plant genetics, plant genetic engineering, and plant molecular marker assisted breeding. So this is the main area of the green biotechnology which we are observed. So biotechnology to make plants pest and drought tolerant. BT cotton, we know well-known the transgenic plants, that is modified, the genetically modified organism or genetically modified plant, that is a BT cotton, is the best example of bollworm tolerant plants. It is also a transgenic plants. We are observing the huge agricultural practices we are observing in the entire world. So that is the Bacillus thuringiensis, gene transfer mediated plants. So that is the BT cotton. So this is belongs to the green biotechnology. And the second one we are observed, that is a red biotechnology. So red biotechnology, it is concerned to the medical sciences. It is related to the medical sciences. The development of innovative drug treatment. Nowadays, we are facing lots of problems in last 2 years in the pandemic situations just because of COVID-19. Okay, so that's why the red biotechnology, which is very useful for the concern related to the medical sciences and the development of innovative drugs and treatments. The main application of this red biotechnology, which useful for production of the vaccines, production of the antibiotics. We know that the first antibiotic, that is the penicillin. We know that that is a wonder drug, which is first discovered by Alexander Fleming, which treat even though the 19th century, in the 19th century, from the discovery of the Second World War, the wounded soldiers is treated with this penicillin. So this is first antibiotic. For nowadays, the use the population is increased. So the population needs so many antibiotics. So that's why the production of antibiotics and also regenerative therapies, number of therapies, especially nowadays, gene therapy is one of the new approaches in the biotechnology field. So gene therapy and stem cell technology, which is also very important for the production of the new organ in the human beings. So which organ is needed in the future generation and future this stem cell technology is very helpful for the production of the new organ in. So these are the few applications of the red biotechnology. And the next one is the blue biotechnology. So the blue biotechnology, which is use of the sea resources, oceanic environment, which is useful for oceanic environment, especially in marine and also in the freshwater organisms to create products and industrial applications. So that is we can call as blue biotechnology. Okay, in the oceanic environment and also the aquatic environment, freshwater, fresh aquatic environment. So the products of the related to the oceanic and also the freshwater, which is related to the blue biotechnology and white biotechnology. The next white biotechnology. This white biotechnology is applied to the industrial process. The production of the amount of the enzymes and industrial catalysts. We know the catalysts, very useful for the insist or the promoting of the what we can say that promoting of the chemical reactions. So and also uses of molds, bacteria, yeast, and microbial organisms also using for the product, number of new products and various goods. For few examples for white biotechnology. And the next one is yellow biotechnology. Yellow biotechnology, which is related to the insects. It is also refers to the use of biotechnology in food production. Okay, that is yellow biotechnology. And gray biotechnology. Next one is gray biotechnology. I hope you are follow my class, my dear friends. Are you follow my class?
Yes, sir. Yes.
Okay, thank you. So in this gray biotechnology, which we are applications of the biotechnology in the environmental applications, especially the maintenance of the biodiversity and the removal of the pollutants, number of waste, hazardous and pollutants which they are causing of the so many harmful effects of the human being and also the environmental problems. So that's why the gray biotechnology, which deals with how to control the environmental pollutions and how to control the environmental hazardous and how to control the biomedical, especially the environmental conditions. And the next one is the brown biotechnology. This brown biotechnology, which is related to the management of arid zone, especially the least amount of the water conditions or water content is available. So the scarcity of the water and also the desert environment. So this brown biotechnology, which is very useful for the drought tolerance and drought resistance, especially production of the drought tolerant seeds and resistant seeds, and especially resource management. Okay, the creation of the agricultural techniques suited arid landscapes. We know that arid landscapes in our Hyderabad, very nearer to the Patancheru, ICRISAT is situated. This ICRISAT is research on the semi-arid zones, So that's why in that area, the brown biotechnology, which is very useful in that ICRISAT like institutions. And there are a few examples of the brown biotech. These are, and violet biotechnology. This violet biotechnology related to law, ethical and philosophical issues around the biotechnology. We know that the intellectual property rights also, so many problems we are facing nowadays. So these ethical issues, law and ethical issues, which is related to the violet biotechnology. And the last one is the dark biotechnology. So this dark biotechnology, which is related to the bioterrorism. We know that. So biological weapons, they are using biological weapons like microorganisms, they are used. This SARS-CoV-2, it is also nothing but this bioterrorism. So in this scenario, this dark biotechnology, which is useful for the microorganisms and toxins to cause diseases and death and disability, these are all deals with this dark biotechnology. And now we will go into the actual topic, that is applications of the biotechnology. Many ways of the applications, many applications related to this biotechnology, that is medicine, agriculture, industry. Okay, in this medicine, we will going to discuss about the biopharmaceuticals, gene therapy. It is one of the most important area nowadays, and pharmacogenomics and genetic testing. These are all related to the medicine and agriculture. In our India, majority of the people, the population, they are depends upon the primary sector, that is agriculture. 65% of the population nowadays, they are depends upon the agriculture. So in this scenario, this agriculture is also one of the most important area, which is applications of the biotechnology in the agriculture, which is very, very useful for the production of the modified crop, that is the genetically modified organism, GMOs, and biofuels, and plant and animal reproduction, and biofortification. Biofortification is one of the important area. Biofortification and antibiotic, production of the antibiotics and abiotic stress resistance crops, which is very useful for production of the agriculture sector. And environment, we know that the environmental is also one of the greatest cause of so many hazardous, and also which is useful for the natural resources, especially the harmonious, sustainable resources is very, very important for the future generations. But in our today's, we are observed ozone depletion, greenhouse effect, okay, global warming. So these are the problems which we are observed in the environmental problems, and also environmental pollutions, which is also related to this environment. In this scenario, the biomarkers, using of the biomarkers and bioenergy. Before this bioenergy, we know that especially the first one is the renewable energy resources. How to utilize this renewable energy resources, and also the bioenergy is also very, very important for how to utilize in this context. And bioremediation, it is also related to the environment. This bioremediation, we are using microorganisms, especially mycoremediation. Mycoremediation means especially the mycology related to the fungi, fungal organisms, which they are used for the useful for the treatment of the environmental problems. And phytoremediation, which we are used in plants, especially the number of plants, blue-green algae, and the plant-related organisms which they are used in the bioremediation process. And microbial remediation, bacteria, yeast, and some other, even though some of the viruses also used in this bioremediation process. And biotransformation, which they are all related to this environment. And one of the most important sector, that is industry. So industrial sector is a secondary sector in our in our global scenario. So this industrial sector, first one is the food processing industries. In this food processing industries, fermentation process, protein engineering. So these are sectors which are applicable of the biotechnology. And present recent development, one is the Human Genome Project and a 3-parent baby. It is one of the beautiful topic which we will going to discuss today's class. And GM mustard, genetically modified mustard, Brassicacea family members, and gene therapy. So how to use this gene therapy and stem cell technology. These are all recent developments in the biotechnology, in the field of biotechnology, by the uses of the genetic engineering process. And government policies also one of the important area of the biotechnology field, that is NBDS, National Biotechnology Development Strategy from 2015 to 2020, and National Biopharma Mission. These are the government policies which implement in the biotechnology field. So first field, that is the medicine. So in this medicine, biopharmaceuticals, biopharmaceuticals are also known as biological and medical products. Biological or biologic is any pharmaceutical drug products manufactured? Okay. Am I audible? Madam, am I audible?
Yes, sir. Yes, sir.
Yes, audible.
Yes, sir.
Madam, am I audible? Vinapడుతుందా మేడం?
Yes, sir. వినిపిస్తుంది సార్. వినబడుతుంది సార్. You can proceed now.
హలో
You can
మేడం
carry
వినపడుతుందా?
on, sir.
నాకు వాయిస్ రావట్లేదు.
వినబడుతుంది సార్. సార్ వినబడుతుంది.
మేడం వాయిస్ వస్తుందా? వాయిస్
వస్తుంది 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 సార్.
నా వాయిస్ వినబడుతుందా మేడం?
వినబడుతుంది వినబడుతుంది.
Right. So in the field of medicine, so many applications are there. So first one is gene therapy. First one is gene therapy. Right. So first one is gene therapy. Gene therapy is the therapeutic delivery of nucleic acid into patient cell as a drug to treat disease. Gene therapy is the therapeutic delivery of nucleic acid into a patient cell as a drug to treat disease. So this is called gene therapy. Is it clear? Gene therapy. Gene therapy is nothing but the therapeutic delivery of nucleic acid into a patient's cell as a drug treat disease. So this we can call as gene therapy. The next one is the pharmacogenomics. Pharmacogenomics. This pharmacogenomics is the technology that analyzes how genetic makeup affects an individual's response to drugs. It deals with the influence of genetic variation on drug responses in patients by correlating gene expression or single nucleotide polymorphism with a drug's efficiency or toxicity. So this is a pharmacogenomics. Okay, pharmacogenomics is the technology that analyzes how genetic makeup affects an individual response of drug. So this we can call as pharmacogenomics. Okay, in this genetic testing. So the next one is the genetic testing allows the genetic diagnosis of vulnerabilities to inherited diseases from parents to their offsprings. The diseases is transmitted or is what we can say that transmitted through inherent traits from parent generation to their offsprings or their filial generations. So and can also be used to determine a child parentage, that is genetic, and mother and father, or general a person's ancestry. So this is called as genetic testing. In this genetic testing, the hereditary traits which is comes from mother side or father side. So this is called as genetic testing. Okay. Now, in this image, we are observed that is gene therapy using an adenovirus vector. So we know that the vectors are used for gene transfer in the field of the genetic engineering or biotechnology. So cosmids, plasmids, or even the phasmids also we are used as vectors in the gene therapy. So in this gene therapy, in this image, we are observed this is an adenovirus used as a vector. In this image, we are observed that how gene is transferred or enter into the new cell. Okay. Then next application, that is genetically modified crops, GMO, or genetically modified organism or genetically modified crops in the genetically modified crops. These are used in agriculture. The DNA which has been modified with genetic engineering techniques, a new trait is introduced in such crops which does not occur naturally in the species. For example, in the field of agriculture, BT cotton is the well-known genetically modified crop. So this BT cotton is treated with Bacillus thuringiensis. It is introduced in the cotton to make it pest resistant, especially bollworm. Okay, so bollworm resistance Bacillus thuringiensis is the genes are inserted into these cotton plants. In this BT cotton, which is having especially the special type of genes which are present in this BT cotton, that is cry genes. What we can say that cry genes. So these cry genes are cry1AC, cry1AC, and cry2AB. These are the two genes which are helpful for this pest resistance, especially this bollworm. Okay. Then the second one is biofuels. One of the biggest applications in biotechnology is in the energy production sector. So these are these biofuels. These fuels are environment friendly, which is not harmful to the environment, which is not causes any hazardous, not any problems to the environment. So these are very as well as have high efficiency when mixed with the conventional fuels. We know that very well known about the very well known plants like Jatropha curcas. Okay, Jatropha curcas used as a biofuel, and also the Pongamia pinnata seeds also, or Deris indica. These seeds also used as a making of the biofuels. Okay. Then plant animal reproduction. So in this biotechnology advances, let for specific changes to be made rapidly on molecular level. Through our gene expression or removal of gene. Removal of the gene or insert into the organism. The introduction of the foreign genes, since traditional methods such as cross-pollination in plant breeding techniques from ancient farmers, illiterate farmers also, they are used for cross-pollination, and grafting methods and cross-breeding are time-consuming. So this cross-breeding are consuming process, time-consuming process. In earlier ancient farmers, they are especially identified their color, the size and color and shape. So these are the characteristic features. These are the traits identified and observed with the naked eye, and specially picked from the large amount of the seeds. So this is the one method from ancient. From today, these are the grafting methods and nursery techniques. Grafting methods which are very helpful for the production of the new varieties of the mangoes. In present scenario, number of mangoes and number of variety plants and roses, even the flowering plants also successfully applicable of these grafting methods. And biofortification. Biofortification is very, very important in the nutrition. In our country, in all over the world, in the population, we are observed hidden hunger. So this is very, very important for the for this malnutrition. Number of children they are facing in early child ages, they are loss of their eyesight. Okay, so that's why the hidden hunger, especially for this method, we are production of the golden rice. So this golden rice containing vitamin A, So that vitamin A is the precursor of the beta carotene. Beta carotene is the precursor of the vitamin A. So that's why this biofortification process in the this biofortification process insert of the vitamins, the gene which are useful for the inducing or production of the vitamin A in the golden rice. This is the very variety of the Taiwan type A varieties for the elimination of the malnutrition like vitamin A deficiency. And antibiotics. Then Hughes say antibiotics is very, very important in the present scenario because of the human beings, they are decreasing of their resistance power. So that's why in the pharmaceutical industry or even though the medical sector, the using of the antibiotics, huge amount of the antibiotics they are using in this scenario. So these antibiotics not only produced from the microorganisms, but also using of these plants in the plant biotechnology, using of these plants also production of the antibiotics for both human and animal use, consumption of the human and animal uses. This antibiotic protein can be expressed through a livestock feed, thereby reducing the expenditure. In previous antibiotics, they are produced very little amount from the what we can say that on the horses and pigs. But nowadays, using of the microorganisms and plant products, and even though the microorganisms like plants and microbes related to plants and related microbes like bacteria and yeast and some of the viruses also. And antibiotic stress resistance varieties. So since there is very little available of the arable land on the face of increasing population and the very rapid urbanization process, there is a need of develop crops that can be handled these antibiotic stresses such as salinity, that is salt resistance varieties, drought resistance varieties, even though the waterlogging resistance varieties, frost resistance varieties, cold means chilling resistance varieties. So these are all very, very important in the present scenario. And the Israel country has successfully developed such crops which could grow under water stress conditions. So very little amount of the water availability conditions, like in our Andhra Pradesh, especially the Rayalaseema, Kurnool, Anantapur regions, in our Telangana regions also, wherever the water scarcity regions, so these antibiotic stress resistance varieties which are useful for the production of the high amount of the yield. Then environmental area. So environmental area, so we are using of the biomarker. So biomarker is one of the very, very important responses for the chemicals that is help for measuring how amount of the damages in the environment or how exposure of the toxic environment area. So which toxic is harmful to the environment. So identify by the use of these biomarkers. So these biomarkers used for the detecting of the substances, detecting for the pollutants, which are effects of the environment, even though human beings also. Then bioenergy. So this bioenergy, the collective purport of the biogases. We know that biogases contain methane. So biomasses, fuels, and hydrogen are called the bioenergy. Nowadays, bioenergy we can call as bioenergy related to biogas, biomass, fuels, and hydrogen. These are all related to bioenergy. So one of the pioneer examples of the green energy are the which is produced from waste collected from the urban areas. So that is the dry and wet garbages, you know. So this is bio waste. This is also one of the most important of the bio waste management. Okay, so this bio waste, biomass waste, these wastes are helpful to the over the pollutions issues caused in the environment. Madam, మళ్ళీ జాయిన్ అవుదామా? మళ్ళీ స్టార్ట్ చేయండి సెషన్. టైం అయిపోతుంది. పార్వతి మేడం గారు.
Sir.
పార్వతి మేడం, am I audible?
Yes, sir.
Right.
Audible, sir.
Okay, madam. So the bioremediation is one of the important process of cleaning up the hazardous substances into non-toxic compounds. From hazardous to non-toxic compounds, which is called as bioremediation. So bioremediation means from toxic to it is modified from toxic. It is decreases the toxicity of the what we can say that toxicity of the wastes, and also it is converted into non-toxic compounds. So toxic compounds to non-toxic compounds. This process is majority used for any kind of technology. So that is clean up the use of the natural microorganisms. Uses of the natural microorganisms. The natural organisms related to. One second, madam. Okay. The natural microorganisms related to fungal organism. The natural microorganisms related to plants, that is the blue-green algae members. The natural microorganisms related to virus, bacteria, and yeast. So this is related to bioremediation. And by the uses of the microorganisms, by the uses of the fungal, by the uses of the plants, microbes. So these are all used as a bioremediation. And biotransformation. The next one is the biotransformation. The changes that take place in the biology of environment, which are changes of complex compounds to simple compounds. So changes from complex compounds to non-toxic simple compounds. So this is called as biotransformation. So biotransformation, which is related to complex compound to simple compounds. That is, the compounds are non-toxic compounds. Okay, and other way, the round is called the biotransformation process. It is used in the manufacturing sector where toxic substances are converted to bioproducts. So number of manufacturing industries are there nowadays in our country, and also the whole world having the rapid industrialization process. Number of new industries are established. So that's why the non-toxic compounds, it is complex compounds is converted into simple toxic compounds and released to the nearest environment. And this industrial fermentation. So fermentation is one of the oldest and very ancient process. We know that in our kitchen, our mother, and also in ancient human beings, ready to utilizing this fermented juice. That is nothing but toddy juice. And మనం చెప్పుకుంటాం తాటి కళ్ళు ఈత కళ్ళు అని మన వాళ్ళు బాగా తాగుతూ ఉంటారు చూడండి. So that is nothing but the fermented juice. In our kitchen, we know that our mother is making of the idlis and what we can say the dosa and bonda. This is also is nothing but fermented products. Okay, that is in our homemade products. Then industrial area, number of producing the chemicals and food industries and bakeries, especially bakery industries and beverages industries, and some of the paper and textile industries also used for this industrial fermentation process. And the significant development in the moving away from greenhouse gases. Okay, then production of towards sustainable production has also been achieved by this industrial fermentation process. And food processing. In our day-to-day life activities, our agriculture farmer is only produce only raw material like rice, like what we can say the jawar and wheat. These are all raw material from the agricultural field. But the food processing industries, after production of the yield, this yield is converted into number of new products that we can call as food processing. In this food processing, fermentation process also we are applied in the making of the new products. And even though the protein engineering is one of the new area for favorable enzymes of microorganisms which are responsible for the improvement of the fermentation process and produced commercially used large scale, not a little amount, used in the large scale for culturing of the huge tanks, the very large tanks. So that is protein engineering. And the recent developments. Now we will going to discuss the last area of today's biotechnology applications, that is the recent developments. The recent developments, the one of the most important, the human genome projects. So human genome project, it is an objective of the HGP. HGP means human genome product, to produce a blueprint of the sequence of genes and spaces between genes that make up a typical human genome. So the objective of this human genome products is to write or build an artificial human genome with the sophisticated. Okay, so this is one of the important area of the human genome project. So human genome project, which is useful for the right of the genome makeup, which is how, what are the gaps among the genes of the human DNA. And bioengineering tools. These bioengineering tools, the potential benefits of the human genome project, right, to India include providing new solutions of diseases like malaria, dengue, okay, chikungunya. So another area where HGP right can revolutionize healthcare is production of the vaccine development. So the traditional way to developing vaccine is too time consuming and very expensive, very expensive. So one can accelerate means speed up. Accelerate means to speed up the process tremendously from the producing of the viruses synthetically and then use those for vaccine development. Okay, so this is the bioengineering tools. So we can use as a bioengineering tools as a microorganisms for production of the new vaccines. And the next one is Earth Genome Project. After Human Genome Project, Earth Genome Project is also one of the new project, which is an international consortium of scientists which will undertake the project that aims to sequence, catalog and characterize the genomes of every eukaryotic biodiversity on Earth over a period of time. And the time especially related, the stipulated time is 10 years to sequence 1.5 million species in 3 phases. Okay. The EGP means Earth Genome Project will help to create a detailed genetic sequence and reveal evolutionary connections among genus, order, and families that will make up the digital library life. This is the main aim of the Earth Genome Project. So main aim of the Human Genome Project is to write up how many genes are involving in the hereditary process of the entire human being. And we can also modify very easier process in a sophisticated manner. So that is the main aim of the Human Genome Project. And Earth Genome Project which is useful for the entire genome, entire genes or entire living organisms from ancient to present scenario. That is nothing but evolutionary process from genus to families. Then next one is the three parents baby. Have you ever heard this term, three parents baby? Okay, we will discuss. human offspring produced from genetic material of one man and two women through the use of assisted reproductive technologies. So one man and two women involving in the production of the new baby. So this is nothing but three-parent baby. In this specifically mitochondrial manipulation process or the replacement of the mitochondria. These technologies and three persons in vitro fertilization process. IVF means in vitro fertilization process. We know that surrogacy method. Ever heard term surrogacy method? Surrogacy means two parents, one male and one female gender, male sperm and woman ovule is separated in the in vitro fertilization process, and they are going to fuse or fertilization in the in vitro method, and after fertilization the zygote is going to insert another female womb. Okay, so this is the new method we can adopt in this process. That is three-parent babies. Okay. Recently, before 2-3 years back, Amir Khan, they are using this surrogacy method and produce. Madam, I'm unable to share. Please give me host. Madam, Parvati Madam garu. Parvati Madam garu. Okay. Next one is GM mustard. So Brassicaceae member GM mustard. That variety is the variety is Dara mustard hybrid 11. It is a genetically modified mustard hybrid. These hybrids are normally obtained by crossing two genetically diverse plants from the same species. Okay, how to produce this GM mustard, my dear friends? This is one of the new variety which is produced from two genetically diverse plants. Okay, so the hybrid name is Dara mustard hybrid 11. Okay, so the first generation offspring resulting from it has higher yield than what either of parents is individually capable of giving, but there is no naturally hybridization system in mustard. Unlike in say cotton, maize, tomato, like these number of hybrids plants. But in mustard, there is no natural hybridization system is available. So in this process, this is because its flowers contain both the female, that is the pistil, and gynoecium, and male, that is the stamens, androecium, parts of the reproductive organs, making the plant naturally self-pollinating. They are self-pollinating. Then what scientists has done to create a viable hybridization system in mustard using GM technology, the resulting GM mustard hybrid produced. So it is claimed 25 to 30% of the more yield than the best varieties of the Varuna. Varuna is related to GM. Varuna is related to mustard variety. But compared to that GM Varuna, compared to natural hybrid, that is Varuna mustard, this GM mustard, Dara mustard hybrid 11 is produced. Compared to Varuna, 25 to 30% of the yield is increased. Okay, currently in the country, this technology is developed by the Center for Genetic Manipulation of Crop Plants in Delhi, okay, in our India. Then gene therapy, stem cell technology. Gene therapy is designed to introduce genetic material cells to compensate for abnormal genes to make a beneficial protein. So through this technique, doctors can treat disease such as cystic fibrosis and hemophilia, muscular dystrophy, sickle cell anemia, and large B-cell lymphoma. Are all genetic disorders. These are all the genetic diseases. These diseases is inherited from parents. So this type of disease also is treated with the help of gene therapy. Then next one is the stem cell therapy. So stem cells have the ability to build every tissue in the human body. Previously, Savita Madam Garu has told totipotency. So the totipotency is the ability of the cell. The one cell is produced, the ability to produce entire the plants. So the totipotency is only the ability of the plant cell, but human cells or animal cell not having that ability. The totipotency is ability related to plant cell. But in this regard, in this scenario, human cells or the animal cells is also rebuilt or rebuild the entire tissue by the help of these stem cells only. So this is the great potential for the future therapeutic uses. Okay, so the tissue regeneration and repair process by the uses of the stem cell therapy. They are broadly classified into pluripotentism. Pluripotency means, pluri means number of organs. Pluri means poly, number of organs is produced by these pluripotent stem cells. And multipotent stem cells. Multi means number of. Multi means so many times. Okay, these pluripotent systems so named because they have ability to differentiate into cell types in the body, while multipotent stem cells can become only cells of a particular germ line or only cells of particular tissue. So multipotent stem cells only related to particular tissue or cells, but the pluripotency means number of total. So there are number of new critical organs like liver, heart, lungs, kidneys. This type of organs is also produced by these pluripotent stem cells. Okay, these are the difference between pluripotent stem cells and multipotent stem cells. And what are the government policies? What are the government policies implementing for this biotechnology applications? Number one, National Biotechnology Development Strategy 2015 to 2020, that is NBDS. And the second one is, the mission will be, the second one is, I will tell you the next second slide. This is the first slide related to National Biotechnology Development Strategy, which is became from applied from September 2007, provided insight into the enormous opportunities. After this, NBDS, National Biotechnology Development Strategy, on December 2015, with an aim to establish India as a world-class biomanufacturing hub. Biomanufacturing hub. It intends to launch major mission backed with significant investments for the creation of new biotech products, creating strong infrastructure for research and development. R&D means research and development, and the commercialization and empower India's human. The mission will be implemented by Biotechnology Industry Research Assistance Council, that is the BIRAC. The mission essential and investment of over 1,500 crores by Government of India for 5 years with 50% cost for the program coming from the World Bank loan. Then key elements of the NDBS are to revitalize the knowledge environment at par with the growing bioeconomy, focus on biotechnology, tools for inclusive development. Then the strategy, what are the strategies for implementing of this NDBS? The build a workforce and improve research facilities in basic disciplinary and interdisciplinary streams of scientific studies, and nurturing innovation, translational capacity and entrepreneurship, and ensuring a transparent, efficient and globally best regulatory system and a communication strategy, and creating a technology development and translation network across the country with global partnership, making India ready to meet the challenge of achieving $100 billion by 2025, launching four major mission: healthcare, food and nutrition, clean energy and education sector, and strategic and focused investment in building the human capacity by creating a life sciences and biotechnology education council. And National Biopharma Mission, this is the second strategy. Second, it is an industrial academia collaborative mission for accelerating discovery research to early development for biopharmaceuticals. The World Bank assisted Innovative in India, I3. We know that iPhone, I1, I2, I3, you know. So Innovative India, III, Innovative in India, I3. Okay, this is the program under this mission aims to create an enabling ecosystem to promote entrepreneurship and indigenous manufacturing in the sector. The focus of the mission is to develop new vaccines, biotherapeutics, diagnostics, and medical devices to address the rising burden of disease. So just because of this technology, just because of this biotechnology, this bring isolated centers of excellence, academia together, enhance regional capabilities, and strengthen the current bio clusters network in terms of capacities as well as quantity and quality output. Deliver 6 to 10 new products in the next 5 years. And create a several dedicated facilities for next generation skills. And to develop platform technologies for product validation, link institutions to strengthen clinical trial networks, promote partial de-risking for novel products, and build capacities in emerging areas such as biotechnics, bioinformatics, etc. And biosimilars for cancers, diabetics, rheumatoid arthritis, and medical devices and diagnostics. So before going to conclusion, nowadays, before 2 years, from 2019 to 2022, tremendous changes we are observed in the global scenario. Just because of one microorganism, SARS-CoV-19, we are facing lots of problems. We are losing valuable lives. Even though young people also, they are lost their lives just because of this microorganism, that is the virus SARS-CoV-19, that is COVID-19. So just because of this biotechnology, implementing of the genetic engineering technologies, we are producing. Our India, we are proudly say that our India is producing high amount of the vaccines and also the exports to the helping for the other countries. Other countries also, they are depending upon our biotechnology field of the biotechnology department of our nation. So that's why biotechnology was assessed with respect to its application. The assessment shows that biotechnology is applicable in the environment, in the field of the medicine, in the field of the agriculture and food processing industries also. It was observed that the areas and scope of application will continue to broaden as science advances. It was concluded that biotechnology research work should be directed towards the risks and challenges and identified, especially in agriculture application. Okay, so this is about the today's class, my dear friends. And before going to conclusion, I would like to acknowledge one of the prestigious institution, government, Pingali Government College for Women, Autonomous, Warangal, the chairperson, Dr. G. Rajareddy Garu, today's seminar, principal, Pingali Government College for Women, Warangal, and vice principal, Dr. G. Swasini Madam Garu, Asst.Professor, Department of Zoology, and controller of exams, Dr. Ramakrishna Reddy Garu, Government, Pingali Government College for Women, Warangal, and today's convener, Dr. D. Parvati Garu, Head Department of Botany, and co-convener, K. Sarita Madam Garu, Assistant Professor of Botany, Pingali College, Government College for Women, Warangal. So this is today's my class. Please, any queries, please you ask me. Any queries?
Thank you, Tirupati sir. I request you to our Vice Principal Madam, Dr. G. Suhasini Madam, please speak about this seminar, sir. Some few words, ma'am.
Thank you, madam. Thank you. On behalf of Pingali Government College for Women, Warangal Autonomous, I thank the resource persons, Dr. P. Tirupathi sir, Assistant Professor of Botany, GDCW Jagithyal, and P. Savita Madam, Assistant Professor of Botany, GDC Lakshadpet, for delivering elaborate and a very useful and also covered various aspects which are being used in our daily lives and also their applications or recent developments on the tissue culture and recent biotechnology applications. Thank you very much, sir, for enlightening our students on various aspects. And I also congratulate the convener, Dr. Parvati Madam, and the co-convener, Sarita Madam, for making it a grand success. And also Sandhya Madam, Sandhya Rani lecturer in Botany, for coming over to this seminar. And this type of seminars and the exchange of extension lectures by different faculty will definitely be useful to the students, as it is an exchange of views and also different methodologies being used by different teachers. Thank you very much, madam, for conducting this type of seminars, and I thank our principal sir, Dr. G. Rajareddy Garu, for giving permission to conduct, and also to encourage all the faculty to conduct this type of seminars. It's nice to see Tirupati sir after a long time. We met in our the OC.
Thank you very much, Swasini ma'am.
Swasini Madam, thank you very much for giving for a wonderful words regarding of today's seminar. And nice to meet you, madam. Nice to see you in the seminar. Thank you very much, Parvati Madam Garu.
Okay, sir. Madam, please give the feedback for Tirupati sir, ma'am. Sarita ma'am.
Okay.
Sarita madam.
feedback, ma'am.
Hello.
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 Harita madam.
Sarita madam. Please give the feedback, ma'am.
And Vice Principal Dr. G. Suhasini Madam for encouraging to conduct this national seminar. And we are also thankful to Savita Ma'am for accepting our invitation and delivering for lecture on tissue culture and tissue culture techniques and applications. And also we are thankful to Padala Thirupathi sir for delivering the lecture on applications of biotechnology and branches of biotechnology. Sir has given an elaborate lecture on applications of biotechnology and covered recent aspects like genome projects and three parents baby and government policies and strategies regarding to the biotechnology development in India. So students are very much interested, and I think they learn much from this national webinar. And so I thankful to Dr. D. Parvati Madam for giving this opportunity. Thank you, sir. Thank you, Ananda. Ma'am.
Thank you, Professor. Thank you, Suhasini ma'am. Once again, thank you. Thank you, Sarita ma'am.
Okay, madam. Thank you.
Thank you, Suhasini Madam. Parvati Madam.
Thank you, Sarita ma'am.
Parvati Madam.
Sarita ma'am, thank you very much, ma'am.
After
Thank you,
5
madam,
minutes,
for
shall we start
involving
our
the
session?
team.
No,
Madam.
ma'am. It's okay. Sarita, chalinga.
వద్దు అబ్బా, నాకు సాటిస్ఫాక్షన్ ఉండదు.
మళ్ళీ ఒకసారి పెట్టండి మేడం. నెక్స్ట్ టైం మళ్ళీ కండక్ట్ చేస్తే సరే.
అయ్యో, ఓకే ఓకే. కానీ మళ్ళీ ఒకసారి లింక్ షేర్ చేస్తా, మళ్ళీ ఒకసారి జూమ్ ఓపెన్ చేస్తే అయిపోతుంది.